Hello, and welcome to New Scientist Weekly, your curated selection of the week's science stories. I'm Timothy Revel in London.
And I'm Christy Taylor in New York. This week on the podcast, we've got a triumphant return of moon samples from China's Chang'e 6 mission, a dangerous new strain of mpox in the Democratic Republic of the Congo, and more evidence of just how community-minded Neanderthals were.
Plus, the quest to build an algorithm that appreciates the beauty of chess, not just how to win. But first, this week, a team in the US unveiled a new kind of gene editing called bridge editing that has got us all excited. Some researchers are claiming bridge editing could even be more powerful than CRISPR gene editing, which has already revolutionized biology. Michael LePage, our resident expert on CRISPR, could this really be true, as good as it seems?
Well, possibly. So it's very early days, and at the moment we still don't even know if bridge editing works in human cells. But on paper it looks really promising because it can add or delete pieces of DNA of pretty much any size, whereas CRISPR is limited to making much smaller changes.
Okay, so talk us through this. That sounds like quite a big difference, but why is that so good?
To start with an example, let's say you've got thousands of people who are ill because they've got mutations in a particular gene. Now, it's already possible with conventional gene therapy to add an extra copy of that gene using viruses. The trouble is you can't control where that gene ends up, and if it ends up in the wrong place, the worst case scenario is cancer. Now, in recent years, we've developed these CRISPR-based tools called base editing and prime editing that can fix specific small mutations. So that's much safer because you're just fixing the mutation that's the problem. However, if you've got thousands of people who all have different mutations in the gene that are making them ill, you're talking about creating thousands of different treatments that at the moment will each require individual approval from the regulator. So that's just a non-starter. What we want to be able to do is to add an entire gene in exactly the right spot in the genome so it's perfectly safe, but the same treatment works for everyone. And that's exactly what we think could be done with bridge editing.
I mean, I'm sold. This sounds really important and necessary in a lot of ways. How does it work? Why might bridge editing be able to do this when CRISPR itself can't?
So what makes CRISPR so powerful and useful is not just that it can make changes to the DNA. Lots of enzymes can do that. It's that we can tell CRISPR exactly where to make those changes. CRISPR proteins join up with a bit of guide RNA and they seek out a site in the genome that matches part of the guide RNA. By changing the guide RNA, we can essentially tell the CRISPR to target any site we want. But if you think about adding an extra piece of DNA to the genome, you also want to make sure that you've got the right piece of DNA. So you want to specify that extra piece as well. And that's exactly what you get with bridge editing. The guide RNA specifies not just one target like CRISPR, but two targets. It specifies the site to alter and the DNA to add. And that's the reason it's called bridge editing, because the guide RNA physically links those two bits of DNA together before the actual editing happens. It forms a bridge between them, if you like.
That sounds like something that's pretty complicated to just stumble across. So how did researchers go about figuring out this might be a possibility?
Well, our genomes are full of these genetic parasites called jumping genes. These are genes that code for proteins that basically cut and paste the jumping genes from one place in the genome to another, which means these are basically gene editing machines. And there are thousands and thousands of different kinds of these things that cut and paste themselves in different ways. So bridge editing is a mechanism used by so-called IS-110 elements, and these are genetic parasites found only in bacteria.
So if these are found in bacteria, I know there is some doubt about just how well this might work in humans. Is that where this comes from?
Yes, exactly. This technique's only been tested in bacterial cells so far. But the thing is, even if it doesn't work in initial tests in human cells, it's likely that in time the system can be modified so that it does work in human cells. And the bigger picture here is that lots of teams around the world are trying to create new gene editing tools capable of the kind of changes we've been talking about. And the field's advancing really fast. So I've got absolutely no doubt that these efforts are going to succeed. Whether bridge editing is the key is impossible to say at the moment. It might turn out to be a transformative technology like CRISPR. Maybe we'll find something even better. Who knows?
China's Chang'e 6 spacecraft is back on Earth this week, and it has souvenirs from its visit to the moon. The probe landed in Mongolia Tuesday, filled with samples from its mission to a crater by the moon's south pole. Hopefully that's about 2 kilograms or 4.5 pounds of samples. This is a landmark mission, both for China and humanity's understanding of the moon, and we've got reporter Leah Crane here to explain more. Hey, Leah.
Hello.
Okay, so what are these samples? Where did they come from, besides the moon, obviously? And don't we already have enough chunks of moon from the Apollo missions?
So the big new thing here is that these are the first samples we've brought back to Earth from the moon's far side. And that's really hard because there's no way to do direct communications to the far side of the moon, so it's tough to land spacecraft there, and then it's tough to launch them back off again. The samples themselves, like all the samples from other worlds, are just canisters of dust and pebbles, but they could help us figure out how the far side of the moon differs from the near side, which we know much more about.
What kinds of differences could we see then? Is there a specific kind of data that the scientists will be looking for in these rocks?
Yeah, so they're geological differences. These samples come from inside a big crater, so they could help us date that crater more precisely and learn more of the specifics on how it formed, which could help us learn a lot about other similar craters. It could also tell us about water ice in the area, which is important for potential future explorers for all the obvious reasons that water is important for humans.
I have to say, I'm super excited about all of this, seeing the rocks and the pebbles come back. I just think it's so exciting that we're able to do that. But I can't also help think of another sample return mission that we're waiting on at the moment, and that's NASA's Mars sample return. That's looking a bit dicey, or that it might be delayed now. As a frustrated onlooker, why can't NASA just do what China has managed to do so successfully with the moon, but with Mars? Isn't it easy?
Mars sample return and lunar sample return are totally different beasts. On average, Mars is almost 500 times farther away from Earth than the moon, so sending anything there and back is orders of magnitude harder, it takes way longer, and it's also way more expensive, which is what NASA's dealing with right now. It's also, for reasons aside from distance, harder to land on Mars than the moon, engineering-wise, because of Mars' stronger gravity and thin atmosphere. And all the same issues apply twofold if you want to come back too. So if you think of it, if sample return from the moon is a small step, sample return from Mars is like an Olympic race. There's a reason nobody's done it yet.
Small step or giant leap, maybe?
Giant leap and then a marathon, which that sounds like a lot. All right, well, I guess one of those other missions that people have been talking a lot about is the US's Artemis program, which is looking to get humans back on the moon. I mean, I'm excited about bringing rocks back, but seeing humans on the moon again, that would be just take it to another level, don't you think?
Yeah, I think most people would say that there's something just quite special about seeing other humans walk on another world that can't quite be replicated by we brought some rocks back and we're doing cool science.
However excited I try and get about it.
Yeah, but this is a precursor to that.
Yeah, well, what else is China planning for missions in the next decade? You know, what are their long-term goals? Are they particularly different from those the US or other countries might have?
The long-term goals are pretty similar. I'm pretty sure the next big exploration missions on the books for China are Chang'e 7 and Chang'e 8. Very creatively named. Those are currently planned for 2026 and 2028, and they're both also moon landers designed to learn more about the southern reaches of the moon and to prepare for renewed human exploration there. There's also some planned missions to look at asteroids and comets and Jupiter, and a sample return mission to Mars, but those are tentatively scheduled for the 2030s. Like I said, China's long-term goals are very similar to NASA's. Humans on the moon, new space station, learning a lot about the other planets and assorted rocks in our solar system.
All right, let's take a quick break to remind you of what else there is to learn and love from our team. Speaking of the solar system, this week Chelsea White and Leah Crane, you just heard her now, made everything old new again on Dead Planets Society. That's right, baby. Geocentrism is back. What happens when you make the Earth the center of the universe? Those results are already in your feed.
And on Culture Lab next week, columnist Bethan Ackley brings us a musical treat. She interviews composer David Ibbott, who's crafted an entire symphony using sonifications of data from Mars. How wind, earthquakes, and even magnetic fields can be woven into an immersive, artistic experience. That's coming on Tuesday. A new strain of mpox, formerly known as monkeypox, has been identified in the Democratic Republic of the Congo, with more than 1,000 cases already reported since September. And based on what we know so far, at least, it's looking dangerous. Several hundred people have died, while others are experiencing severe symptoms. Assistant news editor Alexandra Thompson is here. Alex, how serious is this strain?
Well, health experts have warned it's the most dangerous strain we know of in terms of its symptoms, but also how it spreads. So chances are it's already left the DRC and entered neighbouring countries. So the experts are saying urgent action is needed to try and keep this under control.
Okay, so very serious indeed. What do we know about the differences between this strain and the previous ones?
Well, the symptoms are more serious and they're longer lasting, and the mortality rate is also probably higher, but it's tricky to know because we don't have exact case numbers. So there could be mild cases that just haven't been reported, in which case the mortality rate could be lower. But a lot of the cases are also occurring among children who are covered in these horrible sort of full body rashes. And in terms of how it's spread, this particular strain is largely transmitted via heterosexual sex and non-sexual contact, even among people in different households, whereas the mpox outbreak that was called a global health emergency in 2022, that mainly spread among men who have sex with men.
Yeah, and Alex, we're only hearing about this now, but viruses, as we've experienced, usually get their start well before we hear about them. So when was it first picked up by local officials?
It was identified in a small mining town in the east of the DRC in September and has recently been found in cities in the area. The city of Goma, which borders Rwanda, confirmed its first cases on Tuesday, but it may also already be in the DRC's other neighbouring countries like Uganda. Particularly as rainy season's ended, so it's easier for people to travel between places.
All this sounds really concerning, especially for people who live in the region, but also because, as we know, this is a global society and illnesses can travel far relatively quickly. So what's the plan on how to stop this?
Well, it doesn't seem like there is much of a plan yet, unfortunately. Public health experts we've spoken to in Rwanda hope a vaccination campaign that targets health workers and sex workers could help to bring things under control. And this could involve using smallpox vaccines, which we know protect against other mpox strains, but we don't know if they will work against this particular one. But either way, no vaccines have reached the affected region yet, and we don't know when or if they will.
Next, we are going back in time more than 140,000 years to visit a group of Neanderthals. A fossil find has revealed that a Neanderthal child of at least 6 years old almost certainly had Down syndrome. That this child reached this age in prehistoric times implies that they received significant care from their community, or at least that's the claim of the researchers who analyzed the bone. So how exactly can we know this? Assistant news editor Chris Sims is here to explain. Hey, Chris.
Hi, thanks for having me on. So this fascinating insight is based on a single fossilised bone found in a cave in eastern Spain. And isn't it amazing how much one bone can reveal? The fossilised bone is part of the temporal bone from the side of the skull and includes part of the inner ear. It was discovered in disturbed sediment, so it can't be reliably dated, unfortunately. But we do know that the cave was home to Neanderthals from between about 273,000 and 146,000 years ago. So we are talking that kind of time period.
This is such an interesting premise, but why exactly do we think this child had Down syndrome?
Well, the bone was clearly from a hominin, by which I mean someone from the human lineage, but more than that wasn't clear until CT scans were used to create a 3D model of it that could be analysed inside and out. This revealed that the bone was from a Neanderthal, not a modern human, and belonged to a child who was at least 6 years old and probably no more than 10. Being able to look inside the fossil revealed distinctive features in the semicircular canals that are involved in hearing and balance, including one being connected to a neighbouring chamber called the vestibular aqueduct, which is normally separate, and the cochlea, which is crucial for hearing, was especially small. This combination of features is found only in people with Down syndrome.
I feel like this analysis has really got some detective drama vibes about it. It's really amazing stuff. So the finding clearly suggests that Down syndrome has been around for a pretty long time, perhaps for as long as humans. But is there some more we can take from this? What else does it tell us?
It does tell us some more. It adds to the evidence that Neanderthals, far from being brutish and unfeeling cave people, may have routinely cared for other members of their society. We don't know what level of symptoms the child might have experienced, but the inner ear features imply severe hearing loss and reduced sense of balance and equilibrium. People with Down syndrome contribute in all sorts of ways to modern human societies, and the same could well be true in Neanderthal times. However, the fossil features make it likely that the child would have required more care than other Neanderthal children, potentially needing other members of the community to help the parents, who wouldn't have had as much time to gather or hunt food, for example.
And as you say, this is just now one example of several that point to Neanderthals actually being a pretty sound bunch, not really brutish and, as we often think of them, but actually quite a caring people.
Yes, exactly. One of the best examples of that came from the remains of an adult male Neanderthal discovered in Chang'e cave in Iraq. He had a paralysed arm and a badly damaged leg and was probably deaf and blind in one eye. He survived another 10 to 15 years after getting these injuries, to the ripe old age of 35 to 45, so it's likely others helped him get food and move around.
Hey, Tim. I think I know the answer to this question, but have you ever gotten a paper cut?
What a strange question. I have in my life had a paper cut, I can report, though not for a while. I think probably the closest I've had recently was, those kind of cardboard cuts you get when you're opening a package, that kind of thing.
Yeah, definitely an underrated hazard of getting packages in the mail. Well, if you ever wondered, as I'm sure you must have, what kind of paper might be most likely to cut you, scientists now know the answer.
Finally, making the most of that grant money. And how many undergrads did they have to cut for this particular bit of research?
Well, thankfully none. It turns out that you can use ballistics-grade gelatin as a very, very good scientific mimic for human skin, so no dermis was harmed in the making of this research.
I'm pleased to hear that.
But they did try a huge range of paper types, including tissue paper, printed magazines, business cards, and even printed photos, since all of these have different thicknesses and, properties. And they engineered a system that could basically automatically and consistently push each paper onto the gelatin at a consistent angle. And they did try a bunch of different angles, too, to figure out sort of like what was the most dangerous way to handle paper.
Okay, I'm listening. What combo cut was best?
Well, if you want to most easily damage human skin, it looks like your best bet is to take paper that's about 65 micrometers thick with an approach angle of 15 degrees. And the paper in question, that 65 micrometers, is most commonly used for dot matrix printers and scientific journals.
Of course. They cut through.
Cutting edge research.
Cutting edge research. Very good.
It's basically a thickness that is both thick enough to stay intact when you apply pressure to it, but also thin enough to have a really strong impact on a small area of skin. And that's how you get piercing or cutting.
Okay, and is there a point to all of this beyond just amusing podcast hosts?
Well, one takeaway is that children's books should definitely be printed on thicker paper, which they are. This is because children are just, kind of clumsy and could be at a higher risk of cutting themselves if the paper were thinner than it is. So good job, children's books. And maybe even cooler or weirder, the researchers were also inspired to try to make a recyclable knife made from scrap paper. They call it the paper machete, and they could use it to cut apples, cucumbers, and even chicken, at least until it got wet, which I imagine was rather fast because all of those are very wet things.
Yeah, you might need a knife that can resist some wetness, I would say, if you're going to cut any food at all. All right, moving on. I've got one for you about an AI doing something pretty interesting, I think. And I know we report on these quite a lot, but this one is slightly different. Often AIs are being trained to be as absolutely good as possible at a task. But this one has instead been trained to be as entertaining as possible.
Okay, that's fun. What kind of entertainment? Are we talking juggling, three-card Monty, stand-up comedy?
Yeah, all very fun things. In this instance, it is, in fact, chess. So maybe not everyone's number one form of entertainment. But hear me out on this one. AIs, they can already thrash even the best humans at chess. And traditionally, they do this in a really cold, calculating fashion. They calculate a few moves ahead, and then they just pick the best move. But now researchers are looking at how to make an AI chess player that isn't just good, but also performs elegant moves that impress human spectators.
Got it. Okay, so we want to make a flashier AI chess bot. How do you do that?
Well, so the researchers, they started by gathering data on thousands of games from a chess website. And conveniently, when a move is considered brilliant in chess, that's marked in the game data too. So this data set, it had 820 brilliant moves marked in it. They then trained a neural network, which is a type of algorithm loosely inspired by the brain. And that algorithm was able to identify the probability that a particular move was brilliant, according to humans. And it got that correct about 79% of the time.
All right, neat. So is this supposed to help AI chess players replace humans even more effectively then? Not only can they win at chess, but they can be pretty about it?
Yeah, you might think that, but if you ask the researchers, they think this is more a tool to better understand why humans enjoy certain moves, as well as a more fun computer opponent.
All right, we've got another story from the world of automating human stuff then. How would you like a robot that can smile and has essentially human skin on its face?
That sounds like it would be extremely creepy.
Well, let's take a look at a little video and you can tell me what you see.
Okay, so I am looking at a pink blob with surprisingly human-like eyes, and it's kind of grinning at me, and then its face is melting a bit. Christy, what am I looking at here?
Yeah, so I will say I think it's super weird too, but it did kind of grow on me like a skin. But that human skin there, that pink shiny stuff, that is the big advance here. As I said, that skin is basically human skin.
Oh,
really?
Yeah, it doesn't look like human skin, but I'll believe you.
Well, it's not 100. I mean, it's cultured from human skin cells with a collagen scaffold and all kind of slapped on a solid resin base. But it doesn't have...
I've heard that Hollywood treatment, yeah.
But it doesn't have all of the features of human skin. But it does, unlike previous attempts at lab-grown skin, have ligament-like structures, which is part of why it can move so realistically when it's pulled or pushed. So it's very strong and flexible, and the ligament structures also help it stay in place on the robot better. So basically, what you're seeing when it smiles at you is a robotic sort of mechanical pushing that the skin is reacting to.
Okay, so finally we've got a robot that can smile more realistically and slightly creepier than ever before.
Yeah, and that's, I guess, useful because there is this long-term goal of having robots that can communicate with realistic emotion at humans. But this skin does have another cool trait if you're a scientist. If you make a robot face smile for a month straight, you can induce wrinkles in this skin, which could be invaluable for the skincare industry's quest to create and test products for reducing or preventing wrinkles.
Honestly, I think if you look like this robot, you've probably got bigger problems than wrinkles. But, I think everyone should have a look at this video, so we'll link to that in the show notes. That's it for this week. If you like this show and think more people should know about us, which we really hope you do, please give us a 5-star rating or even a review on Spotify and Apple Podcasts.
We've got more fascinating stories from New Scientist on our website at newscientist.com, where the pages are exactly 0 micrometers thick and paper cut free. We'll be back next week. Bye for now.
Bye.
This podcast is produced by OG Podcasts. Find out more at ogpodcasts.co.uk.